Hello, this is Mr. Russell, your AP Biology teacher. Today we're going to be continuing our discussion on biochemistry, and in particular we're going to be looking at macromolecules. So if we look at the word macromolecule, you can break that apart. Macro means large, so we're talking about large molecules. Well, these large molecules are essentially made up of many polymers. If we break this word apart, we're looking at poly, meaning many, mer being unit, so we're talking about many units or polymers. So if we think about polymers, well, how are they made up? They're made up of individual units. So if we look at here's a unit, here's a unit, here's a unit. These little individual units are called monomers. They're going to be known as the building blocks. And these building blocks are held together by very strong bonds, which were covalent bonds. That's how they're going to be linked together. This is going to help organisms to stay intact. When we look at molecules, we're looking at macromolecules. Let's focus right here in this area for a few minutes. Well, macromolecules are formed by dehydration or condensation reactions. If you remember about hydroxides earlier that we talked about, well, the hydroxyl, which is this OH, and the hydrogen must be aligned properly to form water. So here we can see two hydrogens, one oxygen, which forms water. So we've got a hydroxyl and hydrogen that are going to form water. Now, this condensation of molecules and making of a bond, it requires energy. Obviously, a lot of times we're talking about ATP that's going to be involved. And there's things that cause reactions to take place. Remember we talked about enzymes. Enzymes speed up and slow down reactions. Well, we're talking about a dehydration reaction. In order to make a polymer, we're talking about a way to be able to get these two bonds, or this water, out of the system. And when it comes out of the system, this monomer, and this monomer gets linked together to make a longer polymer. So now we have a dehydration reaction, a removal of water. You get dehydrated because you're losing water. Then you get a bond that creates a stronger, longer macromolecule. Now, these macromolecules are broken into individual monomers by hydrolysis reactions. Lysis means to split apart. We know that hydro being water, so the process releases energy. We're talking about putting water back into the system right here is going to break the bonds. The process needs water, needs a hydroxyl, it needs a hydrogen to fill the gaps or fill the bonds of the monomers. So these enzymes here again speed up and slow down reactions. When I think about these hydrolysis reactions or breaking the bonds, I think about being at a pep rally. And maybe there's a big pyramid that's going to be made. Everybody's kind of stacking up on top of each other and they're trying to stay together and they're doing all they can and they're very strong, hanging on to each other. But imagine if somebody came in with a water hose and sprayed them all down. What would happen if we start spraying all these down? They're going to break apart and then that pyramid is going to start falling. So there at that point, and maybe there's somebody hopefully won't get hurt, but you can see that there would be a strong bond that would get broken throughout this process. Therefore, that would be a hydrolysis reaction, breaking the bonds apart, breaking the macromolecules or the polymers back into monomers. Alright, let's look at carbohydrates. Carbo refers to the carbon part of the molecule, and hydrate refers to the water. These macromolecules are mainly sugars. Monosaccharides, think about mono being single, or the individual building blocks, and sac meaning sugar. So we're talking about individual sugars, the building blocks of larger sugars. Polysaccharides are polymers. So you've got your monosaccharides, your monomers, your polysaccharides, your polymers. So polysaccharides are made up from monosaccharides. The chemical composition is a double bonded carbon with an oxygen. So it has 2 times as much hydrogen present in this process. We're talking about sugars. The names usually end in ose, such as fructose, glucose, sucrose, maltose, lactose, hexose. we've got all these different oses. So anytime you see that name, you need to be thinking sugar automatically. Now, if we look at these are going to be the primary energy sources of cells. Your sugars are your oses. Well, carbohydrates serve as the raw building materials for the other three organic molecules. So the covalent bonds between monomers is called a glycosidic linkage. It just means a sugar bond. This is how these are going to be held together. This glycosidic linkage is going to be holding these individual monosaccharides together. So here we've got a glycosidic linkage. So now we've got maltose. That was a result from two glucoses bonding together. Here you can see that dehydration reaction taking place. And because of that, we went from monomer and monomer to two polymers that get formed together, or one polymer made up of two monomers. And here again we can see glucose being the monomer, fructose being a monomer. Now we've got this glycosidic linkage which forms sucrose. Alright, so those were covalent bonding. So if we look at carbohydrates, they can also be sources of stored energy within the cell of organisms. Starch is the energy storage molecule in plants. Glycogen is the energy storage molecule in animals. And we look at cellulose, is going to be a structural component in plant cell walls. Cellulose is the most abundant organic compound on Earth. If we think about things like chitin, what is chitin? Maybe you want to circle this. We can talk about it more in class. This is the exoskeleton of some animals and also fungi cell walls. Now, we hear a lot about lipids. Lipids they're macromolecules that are fats, oils, waxes, and steroids. Most lipids are hydrophobic. Remember that term? Hydro meaning water and phobic meaning the fear of. The lipids are mainly composed of hydrocarbons. All of the hydrogens means lipids have twice the amount of energy as the carbons. So we think about it, there's two main parts of this. If we look at this fatty acid part, remember this is kind of looking kind of phospholipidish, right? If that's a real word. So we're looking at a fatty acid, a hydrocarbon. The number of carbons will be multiplied twice in the living cell. So they also have this 3-carbon, see if I can get that right, they have this 3-carbon glycerol molecule, which is an alcohol to hold the molecule together. Lipids are covalent bonds. They have what's called this ester linkage that's going to be right here. See if I can erase part of this for you. This ester linkage is going to be right in here. It's going to be a covalent bond. It's going to hold the fatty acid and the glycerol together. This ester linkage is a carboxyl. Here's that double bonded carbon and oxygen up here. That's going to be held. It's going to be bonded up here with this hydroxyl, and that's going to help to form and get rid of some water here. It's going to get our ester linkage going so that we got a nice, really strong bond here. Now, if you think about types of lipids, there are going to be several different types. So let's think about triglycerols and triglycerides. These are just your basic fats that we were looking at here. Basic fats and oils. Now, there are saturated fats. We hear about these when we're thinking about foods that we eat. Saturated fats, these are fatty acids that are saturated with hydrogen atoms. That's all that means. So the molecule has no open bonds to put any hydrogen on. These are solid at room temperature. If you look here, you think about your butters and your cheeses and things like that. They're usually associated with animals. They are the bad types of fat when it comes to our diet. These are the ones we're not supposed to be eating. And then you've got unsaturated fats. These have double or triple bonded that could be broken that add more hydrogen to the fatty acids. So these are liquids at room temperature. They're usually associated with plants such as vegetable oil, sunflower oil, or peanut oil. There are also what's called a polyunsaturated fats. They have numerous double or triple bonds, in the fatty acid portion. These are also liquids at room temperature. They also usually form in plants. Now, one last one we're going to look at here is the hydrogenated or trans fats. These are oils turned solid by adding hydrogen and by breaking the double or triple bonded bonds so that in order to transform this into a saturated fat. Now, let's go back and look at our phospholipids for a minute. Remember phospholipids associated with the cell membrane? These molecules replace a single fatty acid with a single phosphate ion. So, let's look right in here at this phosphate group. So here we are, we're looking at, this is the part of the molecule that is considered to be hydrophobic. This is the part that's supposed to be hydrophilic. Philic meaning the lover of water. So remember, we like the polar head right up here and the non-polar tail. This part up here loves water. Loves water. This part down here hates water. Alright, now, so if we're looking at this, it loves water because the phosphate carries a negative charge. And remember that water is polar, so the negative phosphate will be attracted to the positive hydrogen portion of the water. They still have two fatty acid tails, which are hydrophobic, which is what we just talked about. They carry a neutral charge, therefore they're not attracted to water. Now, so if we look at this, we know that the phospholipids, they end up making a bilayer, and they have two layers. If you think about that here, you can see, if you look at a bilayer, here's a layer of phospholipids, and here's a layer of phospholipids. So therefore, we've got a double membrane, or two layers which form that phospholipid bilayer, which is common for all cell and organelle membranes. Now, if we think about waxes, waxes are made by combining alcohols with unsaturated oils. This is an example of lipsticks, and all they've done here is just added color to be able to make the different shades. Now, if we think about steroids, hormones, cholesterols, a steroid has 4 carbon rings with the top ring looking like a house. So here we are. Here's 1 carbon ring, here's 2 carbon rings, 3 carbon rings, and this one kind of looks like the house that maybe you might win in a Monopoly game, or you might be able to buy there. Alright, so what makes them different? What makes individual steroids, hormones, or cholesterols different are the attachment of functional groups. These functional groups help determine the function of the steroid. This is what makes male and female. So if we look at this is also going to be taking place in our cell membranes. Our cholesterol is also a steroid molecule that's going to help keep the cell membrane flexible. And here we can see it right in here. These are all these little yellow things right here. They're going to be considered to be cholesterols. Now, this is going to help our cell membrane stay flexible, and all the membranes need to have some cholesterol in order to be able to stay strong, so that the phospholipid doesn't collapse down on itself. The problem is that cholesterol in excess is bad for your health. When we think about that, well, we store lipids a lot of times as adipose tissue. This is what all animals do. So tomorrow we will continue on our discussion here of biochemistry. So I will see you then.